Okay, so hello guys and welcome back to Excelsior Academy. So in the previous lecture, we saw how in the nucleus, all the proteins, RNAs are getting transported with the help of the proteins imported and exported. Now we also know why with the help of the NLS. Okay, so we have talked about NLS, NES. Okay, so depending on this, we also come to know like what proteins are getting transported to and fro, right? Now, in this lecture, from now onwards, we will be talking about the other organelles, like how the proteins from after translation from ER to Golgi it goes, or to the other organelles like mitochondria, chloroplast, right? So all these will be discussing. And what is important over here is that with respect to your exam, try to remember those signal ka sequence. Okay, that is what comes for the exam. And for example, if I say that this is the tag, or this is a signal for your lysosomes. So what is a tag? So you should know the answer is M6P. What is M6? Is mannose 6 phosphate. Okay, so if this is present, that means the protein is responsible for that particular organelle. Okay, so this is something which you have to understand, and the way by which all these proteins are sorted. So today we are going to discuss the experiments by which the scientists were able to understand. Okay, now let's begin then. Okay, so in addition to the presence of nucleus, eukaryotic cells have variety of membrane enclosed organelles within their cytoplasm. So yes, we know all those organelles are present in the cytoplasm. Now these organelles provide, I request everyone to keep in the mute. Okay. One second. Okay, so we know all those organelles are present, like mitochondria, chloroplast, everything is present. We are just going to understand like how these proteins are going to get integrated into those particular compartments. Okay. So because of this complex internal organization of eukaryotic cells, the sorting and the targeting of proteins to their appropriate destinations are considerable tasks. So for this, the first step in the protein sorting is while the translation is still in progress. So सबसे पहले क्या step होगा? To understand whether that particular protein is supposed to be meant to present in the lumen or in the membrane. Did you get the point over here? सबसे पहला step क्या है? The moment your polypeptide is synthesized, okay, the translation is happening. It should give the signal to the cell that this protein is meant either for the secretion out, या साइटोप्लाज्म में रहेगा, या किसी ऑर्गेनल के मेम्ब्रेन के ल्यूमेन में प्रेजेंट रहेगा, या in the matrix or in the soma. So it just gives you the signal like where this protein is going to be destined. Okay, so proteins are destined for the ER, Golgi, lysosome, plasma membrane, secretion, and from the cell which are synthesized on ribosome. So they are bound to the membrane of the ER. Okay, so slowly with respect to each organelle, we are going to discuss like how it is being done. So as translation proceeds, the polypeptide chains are transported into the ER. So सबसे पहले step क्या हुआ? आपका protein after translation or while getting translated, it goes into the ER. ठीक है? And now from the ER, it goes to the different organelles via Golgi apparatus. Okay. Now let's talk about the ER. Now, so in the ER, so we know it is a network of membrane enclosed tubules and sacs which is present, right? And we know that ER is in continuous with the outer membrane of your outer membrane of the nucleus. Now, it is the largest organelle of the most eukaryotic cells. Now, why? This is just like हम कैसे बोलते हैं हमारी body में largest organ कौन सा है? Yes? Anyone? Which is the largest organ in our body? It is the
Skin.
yes, correct. So in the same way, the largest organelle in eukaryotic cell is the ER. It has got the large surface area, because of which it becomes the largest organelle. Okay. Now we know that the ER have there are two types are there, rough and smooth. So you know why it is rough, why it is smooth, right? I'm skipping that. Now look at the figure over here. So though over here it is written what these are, But in future, if it is coming for your exam or for the interview, they will just mention that this is ER, electron micrograph. Now from that, you should be able to understand that these are your rough ER. Now why it is rough? So you should be able to say that the ribosomes are embedded on this, and that's where it is your rough. Now where on comparison, you see this is going to be your smooth ER. Okay, clear? Now, the ER and the protein secretion. So to understand this, there were certain scientists who, they were working on it to understand. So to understand this, they have taken the pancreatic acinar cells, okay, where they were working about the secretory proteins, okay, the proteins which will be secreted outside of the cell. So uske path ko they were studying with the help of the pulse chase experiment. Now, what does this pulse chase experiment say? Anyone who can explain or who can just give the gist, like how this pulse chase experiment is being conducted? Akshay, would you like to try?
I don't know, ma'am.
Okay, no worries. Anyone who have got this idea, like what is pulse chase experiment? Okay, so in pulse chase, नाम से क्या पता चल रहा है? Like this is for the few fraction of seconds. Chase means you're gonna make a track of it. Okay, so pulse chase experiment में क्या होता है? So they have used the radio labeled amino acids they have used. Okay? Now when you say radio labeled amino acids, it means the certain atoms in the amino acids are being radioactive. Okay? Now when you have pancreatic acinar cells, so these cells were incubated in the media containing radioactive amino acids. Is this much part is clear? Now, it is not for longer time. It is only for shorter times. That is for pulsing. Like itne pulse ke liye tha. Like for 5 seconds, it was incubated in the radio label or radioactive amino acids. Then it was taken out. It was then freshly grown on the media without having any radioactivity. Then again after some time. So what they do is they will have batches. Okay, they will have the batches of these. Let me explain you the experiment first. So let's say that we have taken this pancreatic acinar cell. So in the batches, so I'm going to do everything in duplicates or triplicates. Okay, always remember each experiment which is conducted in the lab, it's always in triplicates minimum. So let's say we have got the control. Okay, let me share a wide screen. I'll get more space. Okay, so here I'm going to have the cells, right? Now here one batch in triplicates, only the controls is there. Control means only your cells without addition of anything, normally growing cells. Those are your control. Now I am going to have the batches. So what I'm going to do is 1 second, 2 second, 5 second, 10 seconds, let us say. Okay, so here also I have got the plates. Now why I'm making circle? Because those are the petri dishes in which you're going to grow your pancreatic cells. Okay, so again in triplicates, it means this particular cell, this particular batch, I'm gonna incubate in radioactive amino acid containing media. Okay, so what I'm doing, I'm incubating for how long? 1 second. Okay, then I'm taking out, washing it, okay, and freezing the cells. Is this much part is clear? Yeah, okay, cool. So in the same way, again I have taken petri dishes over here, incubated for 2 seconds, 5 seconds, 10 seconds. Okay, now in each case what I've done is I have freeze the cells. Now why I have freeze the cells? I don't want them to grow further. Grow further means then there will be changes in the activity inside the cell. Okay, now what I see is that, let's go back. Okay, sorry. So now what I see is this. Let's see over here. Okay. So what I've done, okay, sorry. So they have taken for the 3 minutes. I just explained same thing in the seconds only. So what they have done is, so in each case they have taken, now they're checking. So you observe under the microscope or whatever instruments you have. Okay. So here, if it is radioactive amino acids, then of course you're gonna do is something called autoradiography. Okay. So you're gonna observe here, like where the radioactivity is seen maximum. So what do we see? Let's say these are those cells. So first the radioactivity was seen maximum where? In your rough ER. Can you see? They have mentioned here and here. So these are your rough ER. On the rough ER, you have seen the first radiolabel proteins are present. Okay. After sometimes what they have seen, like 3-minute pulse label, then after 7 minutes, what they have seen is that this radioactivity from ER, it has shifted towards the Golgi. Okay. Then after 120-minute chase, okay, what now we see that there is no radioactivity here and here. Now where it is present? In the vesicle as well as in the outside. Okay. Now did you get the concept of pulse? Like pulse, in a sense, you have incubated for some time, then stopped. Incubated for longer time, then stop. Then again further longer time, then stop. So this gave the secretory pathway of a protein. So what do we understand from this? What is the path here? That after the translation, the protein has gone to your rough ER. Next, it has gone to Golgi apparatus. Next, it has gone to your secretory vesicles. And then after that, it is secreted out. Okay. Now this tells you the pathway followed by a protein. So this is an experiment you need to understand. Whenever in future interview, they're asking you to design an experiment to tell which protein is present where. How do you understand that protein is moving from one place to another place? ठीक है? कुछ भी पूछ सकते हैं. For example, आपसे ये पूछा जा सकता है कि there is a bacteria. पहले उसका एक प्रोटीन यहाँ पे था, दूसरा यहाँ पे है. How do you understand? What kind of experiments you can do? So this experiment using the radioactive amino acids or any protein you can take, and the same experiments can be carried out. Okay. Everyone got the point over here, the experiments? Experiment part is clear to everyone? Tanvi?
Yes, ma'am.
Okay, cool. What about you, Rishita?
Yes, ma'am.
Okay, okay. So this is how they understood, like what is the path followed by. Okay, so this was the example of a secretory protein, and that's how they saw that from rough ER to Golgi to secretory vesicles to the outside. So this is the path they followed, and similarly they have done for the different proteins, and that's how we understood like mitochondria में क्या जाता है, chloroplast में कैसे जाता है. So all those things at the structural level, everything was studied. Okay, so same thing. So these experiments defined a pathway which was taken by the secretory proteins like rough ER, Golgi, vesicles, and the cell exterior. Okay. Now the entrance of proteins into the ER is a major branch of traffic. So that is something, you know, you can have the analogy to the post office, जहाँ पे सारे के सारे letters आपके पहुँचते हैं. And from there the sorting starts. Like okay, if it is for Maharashtra, it will go to the side जहाँ पे उस state के लिए that particular vehicle is meant. Okay, so that is the first address they see. Then in the Maharashtra, then see the next address. क्या वो पूना है, बॉम्बे है, कोल्हापुर है, क्या है? Then making more specific, they read in such a way, and it is delivered to the destination. So just the same analogy you can use over here also. Okay, so the proteins destined for secretion or incorporation into the ER, Golgi apparatus, lysosome or plasma membrane are initially targeted to the ER, as are nuclear envelope and peroxisomal membrane. So what we are saying is that सारे के सारे protein आपके ER में ही जाएंगे. ठीक है? Now, in mammalian cells, most proteins are transferred into the ER while they are being translated on membrane-bound ribosomes. So what does it mean? It means that as soon as the translation happens, now since we have just completed translation, so you know, you have got the idea कि translation कैसे होता है, right? आपका cap mRNA है, poly-A tail है, then just imagine like how these 60S 40 subunit is coming, making a complex, and आपका translation शुरू होता है. The moment translation starts, okay, few peptides are being synthesized. That peptide जो सबसे पहले synthesize होकर आता है, okay, from the N terminal is a signal that this particular protein should be present in the this particular organelle. Let it be for nucleus या कुछ भी हो. So that signal will help in bringing the ribosomes, which is under the translation, to go and get embedded into the ER. Clear? Is it clear to everyone? So what did I just said? Translation is happening. Okay, this is your ribosomes. Translation is happening. The moment यहाँ से polypeptide chain आया, okay, this particular region, this N terminal, okay, this act as a signal. So it's a signal which will be recognized by certain other proteins, for example, SRP. We'll come to that. So they bring this entire complex, mRNA, ribosomes, everything to your ER, okay, and they get embedded over here. Now, once the translation gets over, ribosomes leaves the ER, and hence the rough ER, what we say, it doesn't mean that agar aapka rough ER aisa hai, it doesn't mean that these ribosomes are fixed. No, it is not. The moment translation gets over, this ribosome will leave. Okay, if any other translation is happening, they will come and sit. Then again they leave and they come and sit. So the positions of the positions of ribosomes embedded on the ER is not fixed. Clear? This much part is clear to everyone? Okay, now
Yes,
yeah, good. So overview protein sorting. So you can just read this later. Look at the figure here directly. So what do we see here? The free ribosomes in cytosol. So those are the proteins which is meant to be present like in the cytoplasm, yeah, mitochondria, yeah, chloroplast. So they will be directly synthesized in the cytoplasm and released in the cytoplasm. Okay? So that is free ribosomes in the cytosol. Now if you have got the membrane bound ribosome जो आपके ER पे present है, so that will come over here, right? Now look at the structure. राइबोसोम का कैसे है, ये आपका mRNA हो गया, ये राइबोसोम हो गया. So do you identify like what is happening? आपका 60S नीचे है and 40S ऊपर है. ठीक है? Usually when we study हम ऐसे बनाते हैं ना, 60 and 40 and ये आपका mRNA और ऐसे प्रोटीन आता है. Since this peptide exit, this polypeptide exit in the ribosome is present on the 60S, ये हमें यहाँ पे चाहिए. ठीक है? And these are the translocon जो they will recognize the signal. This red color polypeptide chain is a signal. It will be recognized and it will take the polypeptide inside of the ER lumen. And from here you can decide कि हाँ भाई इसको Golgi में भेजना है या plasma membrane में या endosome से or whatever, depending on the requirement. Is this much is clear? Like how the free ribosomes and the membrane bound ribosomes are acting? This much is clear. This is an overview. Okay, now let's go a little deeper. Stop me and ask me question if you have any. Okay, so in contrast, proteins which are destined to remain in the cytosol or to be incorporated in mitochondria, chloroplast or the nucleus peroxisomes, they are synthesized on the free ribosomes. Now targeting proteins to the ER. Now how this is being done? So proteins can be translocated into the ER either during their synthesis on the membrane bound ribosomes, that is called co-translation translocation. Guys, this is important. Okay? Co-translation translocation. In simple words, it means translation jab ho raha hai, like it is in the process. That time itself, the ribosomes will come and get embedded to the ER, which will result eventually in the translocation, यानी कि polypeptide chain आपका directly ER lumen में जा रहा है. So that process, entire thing is known as co-translation translocation. ठीक है? Or we can have post-translation translocation. It means translation is completed in the cytosol. Okay, now the protein is brought back to the ER lumen. ये दो तरीके हो गए, ठीक है. So mostly in the ER, in the case of the mammalian cells, it is the co-translation, whereas both co-translation post-translations are used in the yeast. अब आपसे पूछा जाए यूकैरियोट्स में क्या होता है, so it is mainly the co-translation. Okay, did you get this point?
Yes, ma'am.
Okay, cool. Now the first step in the co-translation pathway is the association of the ribosome mRNA complex with the ER. Okay, so for this I have got a video, and this so the reference book for this is Cooper. Okay, yeah, so let's watch this video and see. This is the animation they have shown. Okay, guys, are you able to view the screen?
No, ma'am.
Huh? Hello?
No, ma'am. No, ma'am.
Okay, okay.
All protein synthesis in
And what audio is coming?
Audio is coming through.
Yes.
Okay.
eukaryotes begins in the cytosol, where ribosomal subunits come together and begin translating an mRNA near its 5' end.
Yes?
Yeah.
Yes.
We can't see the video.
Oh, that's what I was asking. Now the screen is visible?
Yes,
Yes.
ma'am.
Okay, okay.
Soon after the protein starts to emerge from the ribosome, it may become directed to the endoplasmic reticulum, or ER. It is targeted to the ER if it is ultimately destined for incorporation into the ER, the Golgi apparatus, lysosomes, or plasma membrane, or if it is destined for secretion. In mammalian cells, most proteins enter the ER co-translationally. These proteins have an amino acid sequence called a signal sequence, which is typically located at the amino terminus of the polypeptide chain. Signal sequences span about 20 amino acids and include a stretch of hydrophobic amino acids preceded by a basic amino acid. As a signal sequence emerges from the ribosome, it is recognized and bound by a signal recognition particle, or SRP, consisting of 6 polypeptides and a small cytoplasmic RNA. The SRP binds the ribosome as well as the signal sequence, and in so doing, stalls translation. The SRP targets the entire complex to the rough endoplasmic reticulum by binding to an SRP receptor on the membrane of the ER. GTP molecules bind to the SRP and the SRP receptor. The GTP binding triggers the transfer of the signal sequence from the SRP to a membrane channel called a translocon. Hydrolysis of GTP to GDP leads to the dissociation of SRP from both the receptor and the ribosome mRNA complex. Transfer of the ribosome mRNA complex to the translocon allows the signal sequence to interact with short hydrophobic side chains located in the narrow neck of the translocon channel. This interaction opens the translocon by moving the plug away from the translocon channel. As translocation proceeds, the signal sequence is cleaved by signal peptidase, and the polypeptide is released into the lumen of the ER. Thus, the process of protein synthesis directly drives the transfer of growing polypeptide chains through the translocon and into the ER.
Okay. So did you get it? Actually, that is the entire thing which I'm gonna teach right now. So did you get the overview, like what exactly we were talking about? So is this part clear where they were talking, where they mentioned about SRP, signal recognition particle? So what is the function of signal recognition particle? It goes and binds to the newly synthesized peptide, because those are the signal, right? So it goes and binds to the signal and brings your entire mRNA ribosome complex to the ER. Is this much clear?
Yes, ma'am.
Okay. Okay, so same thing. You can just go through this later. Let me go to the diagram directly. Yeah. So now how this is happening. Okay, so 1 second, did I miss something? So this we covered in the figure, we covered in the diagram. Sorry, we covered in the animation. Okay, so I'll just once explain exactly how it is happening. This video, I don't know whether it is available in YouTube or not because it's a part of Cooper. Okay, so if you have got the book with you, then you can watch. They provide the CD also with that. Okay, so what we have understood from the animation is that mRNA was there. You got your ribosomes 60 and 40. So यहाँ से आपका peptide आ रहा है, correct? Now this peptide is initially is your signal. This will be recognized by your SRP. Now next, what is the step? This is getting recruited to your, ये अगर ER है आपका ER, this is your translocon. ठीक है? Till now we have just mentioned translocon. Later they have got their own specific name for all types of proteins. So please, okay. So these are the translocon. Now in the translocon, what is happening? This SRP has got the interaction or affinity for this also. So it comes and binds over here, right? Bringing the entire ribosome mRNA complex. So now what we have seen was then you need the GTP hydrolysis. So GTP comes and then the hydrolysis happens. So this GTP hydrolysis is required to make your SRP free. Correct? Now, so when the polypeptide synthesis is still going on, as you saw, the signal peptide is actually making interactions with the hydrophobic amino acids. Okay. And then the entire polypeptide chain goes into the lumen. Now, what you have to remember here is that we have got that additional protein here present, and that protein is your signal peptidase. Okay. So, itne signal ka kya kaam tha? To bring the polypeptide into the lumen of your ER. So now the signal has been removed by the signal peptidase. Those are chopped off and recycle into the amino acids. Now your first folding step of your protein is happening in the ER lumen. Is it all good? Understood?
Yes, ma'am.
Yeah.
Yes, ma'am.
Okay. So now coming to the incorporation of your secretory proteins into the ER vesicles. So अभी हमने क्या देखा? Just you can go through also, like ज़्यादा कुछ नहीं mention है. It is the same what we have just discussed now in the animation what we saw. Okay, so here we have talked like how the ribosomes are targeted over there. The ER signal sequence is present. Then how the signal sequence they're nothing but the hydrophobic amino acids. Now do you understand why the signal peptides are hydrophobic amino acids? Do you understand why it is hydrophobic? Always the signal peptide or these sequences are going to be the hydrophobic amino acids. Can anyone tell me why it is hydrophobic? Just now we saw the animation and it was clearly mentioned in that.
It has to get through the membrane of ER. That is the reason.
No, it has to make interactions with the translocon.
Okay.
Translocon, see, that's what, that's what ER is there. You have got the translocon here. Okay. So this is anyway giving you a hollow gap so that the polypeptide chain goes inside. But since it's a signal and उस signal की जरूरत नहीं है आगे, okay? This portion in the ER membrane, you know that these portions are hydrophobic in nature, correct? This is polar, this is polar. So in translocon also, the amino or the translocon is nothing but again your polypeptide chains, integral membrane proteins. There also these portions are your hydrophilic in nature, these portions are hydrophilic in nature, but here these portions are hydrophobic in nature. Since these are hydrophobic in nature, it can only interact with the hydrophobic amino acids only. Correct. So that's why the starting stretches of the signals are the hydrophobic amino acids, so that they can interact. So it's a confirmation that the correct polypeptide chain has been brought to the translocon. Okay, there are certain proteins which does not need the signal, they just synthesize and directly release into the cytoplasm, तो उसको जरूरत नहीं है ER लाने की. So when they try to enter the translocon, since they don't have hydrophobic amino acid stretchers, it won't have any interactions here, hence they can just move out. समझा? Now it is clear?
Yes, yes, ma'am.
Yes, yes. Okay, so these were the people who started all these experiments and all. Okay, so this we have done. Now the protein is present in the ER. Okay, this ER, how it is happening? See, this is what we need to understand. Then how it is happening from ER, it has to go into the Golgi. अब वो कैसे जाता है? If you have studied every time from Golgi, sorry, if this is a nucleus, these are your ER. From ER to Golgi, it always goes like this, vesicles, and they come back as vesicles, right? It's always vesicles. Your protein does not float in the cytoplasm. There is a carrier. That carrier is nothing but the vesicles. So here what we are understanding is how the proteins are getting incorporated into the ER vesicles, whether it is a membrane proteins or a soluble protein, but it has to go through the vesicle to the Golgi, and in the Golgi the further, you know, post translation modification will take place, चाहे उसमें glucose add करना है या उसका structure change करना है, whatever. Okay, so the further modifications are happening in the Golgi and ER में आपका folding हो रहा है. Now what this is what we have to see, like how it is getting incorporated. Okay, is it clear like why the incorporation is important in the ER vesicle? So that it goes to the Golgi apparatus. ठीक है? So translation of free ribosome, this happens, your protein is free. Okay, either this is there which gets translocated, or then you have got this co-translation which is happening. Okay, so what happens is that if it is required, it will directly go into the lumen. Correct? अब उसको ऐसे imagine करो. This is a nucleus, this is your ER. Now the ER, this particular region is gonna bud out, okay, to go into the Golgi. यहाँ पे आपका this translocon is present and the bound membrane mRNA ये आपका यहाँ आ जाएगा. So what is happening? Co-translation, translocation, by the time this bud out and you have got present in the lumen of the ER. And this forms a vesicle, goes to the Golgi. Written में क्या क्या वापस आएगा? The translocon will come back and ribosomes will move out. They will leave the ER. Did you get this part? Anyone? Should I explain again? Yes, should I explain again, guys?
Yes, ma'am.
Okay. Okay, see, I'll go once again, slowly. Okay. So this is clear to everyone, like how the proteins are translated on free ribosomes. This is clear, right? And you get a protein. The only difference what you see is that here again the signal peptide is there. This is again going to be hydrophobic in nature. Okay. If, see, every protein has got two fate, either to be a soluble protein or to be a membrane bound protein, right? Depending on the function. Now, using this protein, if it is soluble, then अगेन इट डिपेंड्स किसके यहाँ पे सॉल्युबल है, ER ल्यूमन में या माइटोकॉन्ड्रियल के इंटरमेम्ब्रेन स्पेस में और इन द मैट्रिक्स, राइट? For example, now what I am trying to say here is that if suppose the proteins are synthesized first, translated first, then again these proteins will be recruited by other mechanisms and it will be taken inside the ER lumen. एक ये तरीका हो गया where this is the post translation translocation. Just now we discussed, right? So this comes under the category of post translation translocation. Okay. Now here if it is ER में present है, ठीक है? यहाँ पे क्या किया है? These scientists they have added the ER vesicles. Why? Because see, this much is clear to you that the translocon is present and to that ribosomes mRNA are coming and binding and then they are releasing the polypeptide chain into the vesicle. Is this much is clear?
Yes,
Yes,
ma'am.
ma'am.
Yeah, good. So that is your co-translation translocation. Okay. अब उसी चीज को अब बस थोड़ा सा एक्सटेंड करना है. क्या ये न्यूक्लियस हो गया, ये आपका ER हो गया, ठीक है? And here let us say that these are your Golgi apparatus. ये ER हो गया है, ठीक है? Now what happens is that it's not that only from here the vesicles will form. Vesicles आपके यहाँ से भी बनेंगे, यहाँ से भी बनेंगे. The only thing is जहाँ पे आपका ट्रांसलोकॉन प्रेजेंट रहेगा, ठीक है? And in the lumen, see ER, ये आपके डबल ऐसे bilayer है, right? So in the lumen over here, if it is a soluble protein, it will come like this. And वही चीज वहां पे bud off होके vesicle बन जाएगा and it will go to the Golgi apparatus. That's it for further modification. That's all. And with this experiment, when these guys were studying, what they found was that if it is a free ribosomal protein and a co-translation translocation wala protein, what they found was that there was a slight increase in the structure. यानी कि free ribosomal proteins were larger as compared to your these proteins. See, they did this experiment and therefore they saw that they are slightly larger than the normal secreted proteins. इसका मतलब क्या आ रहा है? Are you getting the experiments? अभी इसी चीज को मैं अभी suppose lab में कर रही हूँ, ठीक है? Let's come to the lab. In vitro scenario where I have added everything, ठीक है? So I got my free ribosomal protein, let's say for 540 amino acids. ये मेरा free ribosomal protein हुआ. Now in my in vitro experiments, instead of ER, I don't have ER now, right? So what I'm doing, मैं छोटे छोटे vesicles डाल दूंगी ER के. Now this translocon, wherever they are present, they also have the signal peptidase present. Okay, now this is soluble protein, this is soluble protein. I have harvested. What I found was that अगर मैं ER के through soluble protein लाती हूँ, so the length is going to be 440 amino acids. So from this and this, what is the conclusion? That the free ribosomal proteins where ER is not involved, those proteins are larger as compared to these proteins. Why? What is the answer here? It is because the signal is,
Signal peptide.
yes, it is because the signal is being chopped off by the signal peptidase. That's it. That's it. Okay. एक छोटी चीज समझाने के लिए they do so many experiments. Now it is clear. We'll come to like how from ER to Golgi it is going. We'll come to that. Don't worry. Now little bit concept is clear with that animation and this thought.
Yes, ma'am.
Okay.
Yes, ma'am.
Okay. So now the mechanism by which these proteins are targeted to the ER during the translation is now well understood. Now it is all because of the signal sequences. Okay. Again, they are going to be hydrophobic residues. Now signal sequence of a growth hormone they have given. So itna bada, and you can see that it is mainly the hydrophobic amino acids are present. Okay, now they all do also mention the cleavage site of the signal peptidase. So signal peptidase आपका यहाँ cut करेगा. So that's okay. You don't have to by heart कि कौन से हार्मोन का कौन सा signal है, because we have got millions of proteins. So millions of signal sequences is gonna be there. Okay, so that's okay. It just understand that these signal sequences are present and they are hydrophobic in nature. Okay. Now the same thing. So as they emerge from the ribosome, signal sequences are recognized by the SRP particle. Okay, so which consists of 6 polypeptide and small cytoplasmic RNA and all. So this SRP binds to the ribosome as well as the signal sequence, further inhibiting further translation and targeting the entire complex so that it can go and bind to the SRP receptor which is present on the ER. अभी क्या हुआ? SRP ने पहले आपका signal peptide recognize किया. Okay, now this entire thing is brought to the SRP receptor which is present on the ER membrane. SRP receptor के बाजू में आपका है translocon. Okay, so this binding of the receptor, binding to the receptor triggers a hydrolysis of GTP which is bound to the SRP, as you just saw in the animation, right? So now what is happening? SRP is getting released and the signal sequence of your growing polypeptide chain starts going into the lumen of your ER. Okay, yeah, same thing. See, this is what we have seen in the animation, right? Let's go one by one. Anyone would like to try explaining if the concept is clear? Yes. Rishita, would you like to try explaining?
Ma'am, there's a lot of disturbance here, so I'd
Okay.
prefer.
Yeah. Okay, no issue. Tanvi, would you like to try?
Okay, ma'am.
Hmm.
First step is when the signal sequence, that is N-terminal polypeptide, it comes and then SRP comes in, binds with it and go to the ER, ER, and then the insertion takes place, and this hydrophobic amino acid chain that polypeptide has, that is that gives signal to the translocon. Yeah.
Yes.
And and then insertion takes place, and at the last, signal peptidase chop off the extra polypeptide. Yeah, and that's how that is polypeptide is incorporated.
Okay, so what you have to say is, once it starts, see, the SRP leaves this entire complex once there is a GTP hydrolysis. Okay, SRP becomes free for another round. Now, this hydrophobic signal residues, they starts making interactions with the, as if you remember, there is a narrow gap which is present in the translocon. It gets inserted over there. Okay, now this portion gets recognized by the signal peptidase. Here, this is the signal peptidase. Okay, now this makes it free. The N terminal now goes inside of the ER lumen. Okay, so this is how any protein after translation, it gets incorporated into the ER. Abhi clear hai? This is where the protein is going to be soluble in nature. Soluble matlab ER mein present hai. Now just imagine if the protein is meant to be incorporated into the membrane. Like in future usko integral membrane protein banna hai, right? So then how it is going to be? Okay, those not added. Sorry. So I'll just explain here. Though I mean the slides, further slides I didn't make. Okay. So what is happening over here is that the proteins now, the protein. So this is soluble protein. Okay. Now in the case of membrane bound, you saw that the signal is getting chopped off. Okay. Instead of this N terminal coming to this side, what happens is that this particular alpha helices will be there. If you remember, all our integral proteins or transmembrane proteins, they are always the alpha helices. Only in few cases they are the complete beta sheets, right? So these alpha helices is there. What happens is that there is a push, okay? So this translocon will be different. So उसमें क्या होगा? ये जो होगा push होके यहाँ पे निकलेगा आके, ठीक है? By the time there are certain regions which forms the looping over here. Then again this portion will be inserted, then again push. So eventually what is happening in the ER membrane, okay? I have got my N terminal over here, then something over here, then again loop back, come back, loop back, come back, like this. Okay, then this is C terminal. So they all come together and form a transmembrane protein. Thoda sa idea mila because I haven't added the figures. And maybe tomorrow I'll add the figures and we can talk about the membrane protein insertion in the ER. So far good, guys. Did you get like how in the ER the proteins are getting incorporated? Please do remember all these proteins because ये सब exam में पूछे जाते हैं where we talk about the SRP and the translocon. Later we need to remember the names of the translocon. ठीक है? Because it is going to be very specific for mitochondria, for chloroplasts, for the other organelles, right? So that's why you have to remember the names. Right now for ER, we are just saying that it is the translocon. Okay. There is something which I would like to ask. I have given you one assignment regarding the E1, E2, E3 of ubiquitin. Anyone who did this assignment? No, that's bad. Okay, so tomorrow we'll discuss about that also, like which amino acids it is getting bind so that the ubiquitin molecules are transferred to the protein so that it can be targeted for your proteasome degradation. Okay. So yes.
Ma'am,
Yes.
could you repeat that assignment? I wasn't there. I would
Okay.
like to try as well.
Yes, yes. I mean, it's very simple, Shruti. So it was like, so I have already mentioned that there are 3 enzyme subunits which is involved in the ubiquitin protein degradation. So the question was like, what are these enzyme names? Okay, how ubiquitin, okay, ubiquitin molecule is being transferred from E1 to E2 to E3 through directly to the protein. So once your protein is there and it has got ubiquitin molecule attached, this is a signal that it should be recruited to the proteasome where this should be completely degraded and converted into amino acids. So question क्या था, ये ubiquitin molecule कौन से amino acid में present है and from where it is getting transferred from E1 to the polypeptide chain which is meant for the degradation. Those amino acids आपको पता करना है, नाम, function and at which amino acid level it is functioning. That's it.
Okay.
Yeah, because directly this question has come for the exam. Directly, इसमें कुछ logic नहीं था, कुछ नहीं था. Directly this question has come for the exam. Okay, so that was the assignment. Let's see tomorrow. Let's hear the answers. Okay.
Okay.
Yeah, yeah. Anything else? Anyone who would like to ask a question? So today, one second, let me stop.